Okay, so here we are going to discuss protein structures, and this is just the first part. We'll be discussing more about protein structures, but an introduction. So the first thing you have to talk about is hypervariable regions. So one thing we know is that we go from DNA to RNA, and this will give us our proteins. And of course, our amino acids are built upon to make our proteins. So encoding proteins comes from DNA. And so that's where we start with our story because that's where you find hypervariable regions. So this is in the DNA. And hypervariable regions are when you have different amino acids at a single position that are tolerated. Now remember, I told you that you don't want to change the amino acid sequence too much because that will change the proteins. But here I'm telling you that you can have different amino acids at a position within your protein that can be tolerated, and that's called the hypervariable regions. And the tolerated is going to mean that this is a critical region, so it doesn't impact the function of the actual protein. Or if it's conserved substitutions, or if it gives an advantage. An example is our antibodies and our T cells. They have hypervariable regions so that they can bind to many different pathogens, flu versus measles versus tuberculosis, etc. So those are going to be accepted and tolerated. Now the other thing is a polymorphism. Polymorphism is a variant of a gene, what we call an allele. And the variant of a gene is going to occur with a significant frequency in the population. And this can contribute to our individual characteristics, such as hair color, eye color, obvious dysfunctions, such as congenital or genetic inherited diseases, or increased susceptibility to certain diseases. About 1/3 of all loci are polymorphic. And an example of a polymorphism is sickle cell anemia. Sickle cell anemia is a stable point mutation in the hemoglobin gene, and it is at a very high frequency because if you have sickle cell anemia, you can be protected against malaria. Now, a few other things to understand about protein families is there's something called families and there's something called superfamilies. So paralogs are related. So this is where you have same protein family members and these are going to be similar in structure and function. And this is where they have evolved from the same ancestor, but they're considered different proteins. And then you have a concept called divergent evolution. This is where you have a duplicated gene and you have one gene keeps its original function and then one gene changes. And so this is different than a paralog because a paralog has a similar structure and function, whereas divergent evolution actually changes the gene to have a different function. We also have superfamilies, which are very large families of what we call homologous proteins. And the hemoglobin, myoglobin, are examples. And myoglobin and hemoglobin are actually also paralogs. So protein families are going to evolve from some sort of ancestral gene. So here we're showing our myoglobin, and we have our different chains of our hemoglobin. We have a beta chain, an alpha chain, a zeta chain, and a gamma chain. And so for myoglobin, this is going to be intracellular heme protein. This is going to be able to store and transport oxygen to mitochondria. And it's a single polypeptide with one heme binding site. Hemoglobin, on the other hand, is composed of 4 globulin chains, which means it has 4 oxygen binding sites, and is present in the red blood cells. And it's going to transport oxygen from lungs to tissue. And back and forth, of course. Oxygen goes to the tissues, carbon dioxide out, etc. Myoglobin is thought to have evolved from gene duplication of the alpha gene of hemoglobin. And it's thought that the alpha chain of hemoglobin evolved from the beta chain of hemoglobin. Okay, so your myoglobin is shown here. Your myoglobin is monomeric heme. And it's found mainly in muscles. It stores oxygen inside of muscle cells. And during low levels of oxygen, oxymyoglobin will release its bound oxygen. Its secondary structure is going to be primarily alpha helixes. And this kind of structure is very rare. Typically, secondary structures are going to be alpha helixes and beta sheets, and this one has a lot of different alpha helixes. It has one heme prosthetic group, and this is going to be what is going to bind to the oxygen. And the prosthetic group is tightly bound to the enzyme. So histidine is going to stabilize the reaction of oxygen binding to ferrous, which is the Fe+2 form. So O2 binds ferrous. And this is going to be stabilized by histidine. Now, carbon monoxide will also bind, and this has a higher affinity. So carbon monoxide binding a heme is responsible for asphyxiation from carbon monoxide poisoning, and it has a higher affinity, so it outcompetes oxygen. Now, hemoglobin is a tetramer. And tetra means, of course, 4. So it has a tetramer protein, and it's found in erythrocytes, or red blood cells. It's responsible for binding oxygen to lungs, transporting that through the body, as we all know. It has an adult hemoglobin. It has 2 alpha and 2 beta chains. And so you have your alpha chains and your beta chains, and these form the tetramer. And so you're going to have your heme, which has, again, your ferrous iron, which will be able to bind to oxygen, and there are 4 of them. Now, the reason why hemoglobin is so important is because it has a sigmoidal curve. So your myoglobin is what we refer to as hyperbolic. And this means that it gets saturated very fast. And so this is looking at the percent CO2 concentration and the pressure of oxygen. So for your myoglobin, again, your saturation occurs when you're at about this 20 pressure of oxygen, whereas your hemoglobin will not be fully saturated until it's about 50 pressure of oxygen. And so hemoglobin has a sigmoidal curve. Oops. And this is because of the binding properties of hemoglobin versus myoglobin. Myoglobin only takes 1 oxygen. It's easy to saturate a myoglobin molecule, whereas hemoglobin has 4. So the very first oxygen binding is sort of the hurdle. And then once you get that first one binding, the 3 other heme centers will bind. Now, myoglobin versus fetal hemoglobin versus hemoglobin A, or adult hemoglobin. So I'll just quickly go over these. One thing to remember is that fetal hemoglobin has to compete with maternal adult hemoglobin. So fetal hemoglobin will have a higher affinity for oxygen than adult, and myoglobin will have the highest affinity for oxygen, the highest saturation. So first we have O2 saturation on the y-axis. And so this is going to be increasing as shown by the arrow. And then on the x-axis, we have the pressure of oxygen, which also is increasing. So we have them in increments of 20. So we have 20, 40, 60, 80, and 100. And then we have over here, we have our lungs, somewhere between 90. A lot of oxygen out here. And over here in peripheral tissues, we have it somewhere between 20 and about 35. Now remember, your hemoglobin does not want to get saturated in the tissues because it only wants to pick up oxygen when it's in the lungs if it's going to transport the oxygen to the tissues. Now, myoglobin is unique in that it wants to release the oxygen and bind to the oxygen as a storage molecule within tissues. And so our myoglobin will have the fastest saturation. So here's myoglobin. And then we have fetal hemoglobin, again, which has to compete with the mother's hemoglobin. Now, fetal hemoglobin will still have a sigmoidal curve, but it will be a altered sigmoidal curve because fetal hemoglobin doesn't have to get to the lungs. It basically has to pick up oxygen from the mother's blood. So it has a higher affinity for oxygen. And then our hemoglobin A will really not pick up oxygen in the tissues because it wants to pick it up in the lungs. Okay, so hemoglobin A picks up oxygen in the lungs. That's where it gets saturated. Fetal hemoglobin is going to get saturated in the tissues because it doesn't need to get to the lungs. It has to get oxygen to the fetal tissues. And then myoglobin is going to get saturated in the tissues because it wants to store oxygen. So that's where it gets saturated. All right, so developmental variation on our hemoglobin. We have, of course, embryonic hemoglobin, fetal hemoglobin, and adult hemoglobin. Now, embryonic hemoglobin is epsilon and zeta chains. I know they have alpha here, but it's really a zeta chain. I'm very bad at drawing zeta symbols, so I'm just going to write it. So this is really zeta. Zeta is alpha-like. And this is zeta, which is alpha-like. Okay, and then when you get to your fetal, you're going to have 2 alpha and 2 gammas. And then when you get to your adult, your gammas are going to be swapped out for your betas. So within the same individual, you can have different isoforms or isozymes, which they may be called, and at different stages of fetal and embryonic development, different tissues or different intercellular locations. People who have mutations in hemoglobin can have fetal hemoglobin present in their bodies to compensate for mutations in adult hemoglobin, for example. Now, there are also different, hemoglobin's one example, but there are also other proteins that are different isoforms of the same protein in different tissues. And so these isoforms are going to have similar function. So we have denyl cyclase. And this is 3', 5', cyclic AMP synthesis. So these are important because they form cyclic AMP after some hormone stimulation. There are 9 isoforms. There are, different genes in different tissues and they are going to respond to hormones, but their responses can be different in the tissues. So I'm going to put here different responses to the same hormone. Now here we have our M1 and M2, and these are transmembrane domains, which you have your alpha helices in the membrane. And keep in mind that they are going to respond to the same protein, but their response downstream can be different. And here we have invariant C1 and C2 regions, which are important for synthesis of cyclic AMP. Now we also have species conservation with respect to insulin. And so insulin is highly conserved. So for example, you can have bovine insulin, which was used by diabetics before we had recombinant human insulin. And so this is an example of conservation across species. So bovine or cow insulin was used to treat type 1 diabetics. Now for insulin, you have two peptides. You have your A chain and you have your B chain. And these peptides are going to be linked through our disulfide bonds, shown here. And then you have cleavage sites from our C-peptide. C-peptide is something inside of the beta cell of the pancreas that keeps the insulin chains together until it gets released from the beta cell. And so cleavage sites for maturation and release into the blood for your mature insulin are here. Now, one thing to show is that we have some bioengineered insulin, and that's what these little circles are showing. These red circles are showing how the insulin is now bioengineered. One of the problems of giving bovine insulin is that your immune system will eventually make an immune response to the bovine insulin, and then you can no longer use that insulin because it'll get depleted by your immune system. And so these bioengineered insulin, so now we use bioengineered. To treat type 1 diabetes. And also insulin dependent, of course, type 2 diabetes. Now there's a bunch of different translational modifications done to our amino acids. So these are often going to be post-translational. They're going to target or anchor proteins, so they can target proteins. So it can be for secretion, for membrane, for a cell compartment. It can also to anchor proteins into a lipid membrane, for example. The proteins might have to regulate phosphates, and also there can be simple glycosylation. And glycosylation can help proteins fold properly. and this can increase stability. Now, some of the additions, again, are going to be carbohydrates. We can also have lipid additions. So carbohydrates, you can have O-glycosylation or N-glycosylation. This is going to occur in the N-glycosylation. This will occur in the ER lumen. And then O is going to be in the Golgi. It's where O-glycosylation occurs. Now, the glycosylation is going to be highly regulated. Only certain amino acids can be glycosylated, and so therefore only certain proteins that have the amino acids in the right locations can get glycosylated. You also have addition of lipids, and this will help to have the protein interact with the plasma membranes, for example. Polymidylation is an example. Myestrylation is another example. Pomylation is going to occur on internal sulfur groups of cysteines. So here you have a cysteine, and you're going to get a fat attached to it. So fats are very long carbon chains. And for myestrylation, you're going to have a glycine on the terminal, N-terminal glycine is going to have a fat added to it. Prenylation, again, is also going to occur on sulfur groups of your cysteine, and this is an example of a prenylation. And prenylation can link up more than one cysteine. And so these lipid changes are, again, to make it so that the protein can interact with some hydrophobic region, especially with your membranes. And it can be your plasma membrane, it can be your endoplasmic reticulum, nuclear membrane, whatever it happens to be. Now, there are other modifications that are going to regulate. So we have some regulatory modifications, and so phosphates, addition, or removal. So this is phosphorylation. And then this will be dephosphorylation. Is involved in regulation. So adding phosphates or taking away phosphates. Same with acetylation. So again, we can have your phosphate added, and then it can be taken away. We also have an acetyl group that can be added, or it can be taken away. We also have oxidation reactions, hydroxyproline. We have gamma carboxyglutamate reaction, which is carboxylation of glutamate. Remember, glutamate is going to be a excitatory amino acid, and then GABA, it's the carboxylated version, is going to be inhibitory. And then hydroxyproline is going to be important for your collagen and things like that. Now we can also have enzyme forms in different tissues, and one important one to talk about is our creatine kinase. So creatine kinase has a B form and an M form. And so it's going to have brain. B is for brain. And M is for muscle. And throughout the body you have different forms. So for example, in the brain, you have a BB, which is in brain. And you're going to have the MB, which is in heart. And you're going to have the MM, which is in your skeletal muscles. And so after a heart attack, your heart attack can be diagnosed with an increase of MB in the blood. Now, all of these have the same function, slightly different in charges and size. Tissue distribution is going to be different, and development stage is going to be different. So this is just showing your isozymes in the blood. So this is a normal. So you have creatine kinase 3, which is mostly going to be skeletal, and creatine kinase 2, which is your heart, is going to be at very low levels. 24 hours after a heart attack, though, this creatine kinase 2 is going to increase. So we have CK1, CK2, and CK3.